All right, hello 12s. Today we're talking about eukaryotic cells and all their organelles. This video is going to focus on the nucleus. But first, let's talk a little bit about what is a eukaryotic cell. So there are all organisms are made of cells. And there's two main types of cells. There's eukaryotic cells and prokaryotic cells. The big difference between eukaryotic and prokaryotic cells is something called compartmentalization. Compartmentalization. And basically what that means is that in a eukaryotic cell, the places where metabolism happens, where enzymes work, are grouped together into little rooms, organelles, separated by cell membrane, by plasma membrane, lipid bilayer. In prokaryotic cells, there's a cell membrane that surrounds the cell, but all the metabolic processes just happen in a big mess, basically, in a big goo that's just the cytoplasm. Everything is inside one thing. There's no nucleus, there's no nothing, DNA, everything is mixed together. So the advantage of having compartmentalization is you could be a lot more efficient. So this classroom, for example, is compartmentalized, right? There's my desk. This is the area. So when I need to find old exams or if I need to find notes or the computer, I can just find it here. Say you were in this classroom and I assigned you a task where you had to make a poster, okay? You had to make a poster. You had to draw all the components of a cell. Gee, that's a great assignment. Draw all the components of a cell and color it and describe what each of the component, all the organelles in the cell do. So you would need poster paper. You would need to look for that. Well, this is the section of the room that has poster paper. In a eukaryotic cell, this is the part where the poster paper is. So you know where to go and you can get poster paper. In a prokaryotic cell, this poster would just be spread all over the room. And you would have to move around until you just by chance found some of the proper paper. That's what happens inside of prokaryotic cells. The reactants, the substrates of metabolism, of enzymatic reactions, they just have to move around randomly inside a prokaryotic cell until they run into the proper thing that they need to do the thing that they need to do. If you were an enzyme that makes great assignment posters, that might take you some time. It'd take you a little bit more extra time than just going to the part of the cell that has the poster paper. Next thing you would need is some art supplies, some more art supplies, some markers, pens, maybe a ruler, whatever. Well, in our compartmentalized classroom, all the proper markers, say you needed a pink one like this. Is this pink? It's more fuchsia. Are all here. These are the reactants, the substrates of an enzymatic reaction. And here they are. They're all in the right place. You can now come here and color your poster if you need green, if you need fuchsia. But in a prokaryotic cell, these would be all spread out all over the room. How would you find the color marker that you need? Well, you would have to randomly move around the room until you finally found the marker that you wanted. This would take an extra amount of time. So prokaryotic cells, because everything is all mixed together, it's not compartmentalized, are just not as efficient as eukaryotic cells. Hence, eukaryotic cells can grow larger and can carry out those functions more efficiently and better. Converting glucose into energy, for example, as we learned in the metabolic pathways unit in the cellular unit. Eukaryotic cells, for each glucose molecule, produce 38 ATP, whereas prokaryotic cells produce only 2. That's because there is a compartment in eukaryotic cells called the mitochondria that produces an additional 36 ATP. All the enzymes, all the processes are all organized, are all in that one spot, and are able to more efficiently break down the glucose into ATP, whereas it's not possible in the cytoplasm of a prokaryotic cell because everything is too spread out. Okay, so Eukaryotic cells more efficient. So let's talk about some of these compartments. In this video, we're going to be focusing in on this. This is the nucleus. So if we look at a cell, the nucleus is going to be in the center, around to the center. And there it is. There's the nucleus. And so the first thing that we are learning about this compartmentalization, about organelles, is not all of them, but a lot of them have a membrane around them. And so this, like we learned about membranes are semi-permeable. So they allow certain things in, don't allow certain things, let things move. That's how you get that compartmentalization. So you can contain substrates of enzymatic reactions inside the compartment because the membrane won't let them out. It'll let the products out, but the substrates, they have to stay in. It'll only let them in. So this is a nucleus. And the membrane, it has a special membrane. This is the membrane that's around it. It's called a nuclear envelope. Okay, this is the nuclear envelope of a nucleus. And what you'll notice, if I get right in close, is it's these two dark lines. Because the nuclear envelope of the nucleus is a double membrane. So you've got the lipid bilayer, where you've got phospholipids going this way and phospholipids going that way, okay? So you've got one of those lipid bilayers, and then there's another one, another lipid bilayer. And there is a space in between called the perinuclear space. So this is the inside of the nucleus. The inside of the nucleus is called it's called the nucleoplasm, and the outside of the nucleus, the inside of the cell, is called the cytoplasm. So you learn. So that's the cytoplasm. So the fluid in the cytoplasm, fluid and stuff in the nucleoplasm, and the composition of the two are different. There are different things in them because different things can happen. That's the advantage of that nuclear membrane, of that compartmentalization. So other things about the nuclear membrane. So it's a double membrane. And what you'll notice is it has these weird things, these weird structures in it. And these actually have been, using electron microscope, these have been visualized. So they look a little bit like that. They're a complex of enzymes and membranes. So they're a whole bunch of stuff, and they're really big. And basically what they do is they do two things. They're called nuclear pore complexes, and they let stuff in and out. Stuff can come in and out of the nucleus, and only specific stuff. The other thing that they do is they anchor the outer nuclear membrane and the inner nuclear membrane together. There you go. They see them. They're holding them together. Those are the two things that they do. Oh, sorry. That's the two things that the nuclear pores do. They anchor the nuclear membrane, and they let things in and out. And they let things in like nucleotides. RNA nucleotides are allowed in. Those RNA nucleotides will copy, the DNA will copy a gene into mRNA, and the mRNA is allowed out. Ribosomes, the subunits of ribosomes, and we'll talk about what ribosomes are not the next unit, but the unit after. Ribosomes are the subunits are made in the nucleus, in the nucleoplasm, and those then are allowed transported out. There's also the inner nuclear membrane. Has something called nuclear lamina or laminar proteins. We don't see them here, but essentially what laminar proteins do, they just strengthen the inner membrane so that it doesn't fold in or it doesn't damage the DNAs. They're basically like a shell. They kind of create an inner strong shell to protect the DNA, because the DNA is important, right? The DNA, by coding for enzymes, by coding for genes and coding for enzymes, is what controls the composition of all the membranes and all the enzymes inside the cell and outside the cell as well. So we say the DNA, we kind of treat it like the brain of the cell, but it's not smart, it's not thinking, it just produces or it encodes the information for enzymes that the cell will use, enzymes and lipids. So depending on what DNA is read inside the nucleus will depend on what the cell's function will be, what its metabolism will be, what its nuclear membrane will be, what the cell membrane will be composed of, what proteins will be in the cell membrane. All that is controlled by the information in the nucleus. Okay, so it's very, very important. So we talked about laminar membranes, we talked about the pores, we talked about the inner and outer membrane. What else can we see in the nucleus? Well, there's the inside of the nucleus is full of something called chromatin. And chromatin is a combination of DNA. The DNA isn't just alone in here. It isn't just DNA floating around in here, big long strand of DNA. No, the DNA is organized in there, and it is, the DNA is organized with proteins. There are specific proteins that hold the DNA together, that wind the DNA, that hide certain sections of DNA so that it can't be read. And there's certain parts that then unravel the DNA that allow it to be read. When you guys went through puberty, there were hormones that entered your cells and entered the nucleus and unwound certain sections of your DNA in certain cells that targeted specific cells. You got pubes because all of a sudden parts of your DNA in certain cells got unwound that coded for pubes. And all of a sudden, you started making the enzymes and the proteins, the structural proteins and the enzymes that make pubes. That's how the DNA works. That information was there, but it was wound up in the chromatin and it got unwound. I think that's good. So, nucleus, double membrane, things to remember, double membrane, inside is, and nucleolus, let me just say the nucleolus, this is where the subunits of RNA, or of ribosomes are made in the nucleolus. Lots of people ask that. So the subunits are a combination of ribosomal RNA and protein, and we'll talk about that in all the sections. So here we go. Nucleus, center, there's a nucleolus. Center of the cell full of chromatin, which is protein and DNA. It's got the nuclear envelope, which is a double lipid bilayer, intersected by nuclear pores, which control what goes in and out of the nucleus. And there you go.